For most of human history, disease was something people feared but barely understood, something that arrived without warning and stayed without explanation. And when someone fell ill, the question was never why at a biological level, but why at a spiritual or moral one. Because long before microscopes and DNA sequencing, illness was blamed on gods, curses, bad air, fate, or bloodlines. And even when medicine advanced enough to treat symptoms, the root causes remained hidden, locked inside the body at a scale too small to see, until science uncovered a truth that quietly changed everything. The realization that every cell in the human body carries a set of instructions written in DNA that governs how the body grows, repairs itself, fights infection, and sometimes fails. And this discovery reframed disease itself because many illnesses were no longer random or mysterious, but the result of errors, mutations, or inherited variations in those genetic instructions. Meaning that disease was not just something that happened to the body, but something written into it. Sometimes from birth, sometimes accumulated slowly over time. And once humanity understood that diseases could be encoded in genes, a new and unsettling question emerged, one that sounded dangerous and impossible at first. If diseases are written in genetic code, could they be rewritten? Could biology itself be edited, corrected, or redesigned? And this question gave birth to genetic engineering, a field that sits at the intersection of medicine, biology, ethics, and the future of humanity. Because genetic engineering is not merely about treating disease, it is about intervening at the most fundamental level of life, altering the instructions that make us who we are. And in its earliest forms, genetic engineering was crude, experimental, and full of uncertainty, with scientists attempting to insert healthy genes into patients suffering from inherited disorders, hoping the new genes would compensate for the faulty ones. And sometimes these early gene therapy trials produced remarkable improvements, while other times they failed catastrophically, causing immune reactions, cancers, or even death. Reminding the world that changing biology is never simple, yet even in failure these experiments proved something irreversible, that the human genome was not sacred or untouchable, but modifiable. And over time, as understanding deepened and technology improved, genetic engineering evolved from guesswork into precision, especially with the arrival of tools like CRISPR-Cas9, which allowed scientists to locate specific sequences of DNA with extraordinary accuracy and cut, disable, or replace them, turning genetic engineering into something closer to molecular surgery than experimental tinkering. And with that leap came real progress against diseases that had long been considered incurable, because researchers could now target the exact mutations responsible for conditions like sickle cell disease, cystic fibrosis, beta thalassemia, and certain forms of inherited blindness, correcting the genetic errors at their source rather than managing symptoms for a lifetime. And for patients born with these conditions, genetic engineering represented something entirely new. Not just treatment, but the possibility of a permanent cure. A chance to live without the shadow of a disease that had defined every aspect of their lives. And beyond rare genetic disorders, the implications extended into far more common diseases like cancer, heart disease, diabetes, and neurodegenerative conditions. Because while these illnesses are influenced by lifestyle and environment, genetics plays a powerful role in determining risk, progression, and response to treatment. And genetic engineering combined with genetic screening allows medicine to move toward personalization, where therapies are tailored not to the average patient, but to the individual's unique genetic makeup, meaning that instead of trial and error prescriptions, doctors can predict which drugs will work best, which will cause side effects, and which preventive measures are most important for each person. Shifting healthcare from reactive to proactive, from treating illness after damage has occurred to preventing disease before it begins. Yet this power also exposes uncomfortable truths, because genetics does not exist in isolation, and not all diseases are caused by a single faulty gene. Many involve complex interactions between dozens or hundreds of genes and environmental factors, meaning that editing one gene may not produce the desired result, or could create unforeseen consequences elsewhere in the body. And this complexity is especially evident in diseases like cancer, where mutations accumulate over time, hijacking normal cellular processes. And while genetic engineering has enabled revolutionary treatments like CAR T cell therapy, Where a patient's immune cells are genetically modified to recognize and destroy cancer cells, these treatments are expensive, intense, and not universally effective, highlighting that genetic engineering is not a miracle cure but a powerful tool with limits. And those limits become even more significant when considering the ethical questions surrounding the technology. Because while editing the genes of an adult patient to treat a disease affects only that individual, editing embryos or germline cells affects future generations, permanently altering the human gene pool. And this raises questions humanity has never had to answer before about consent, responsibility, and long-term risk. Because future generations cannot agree to genetic changes made today, yet they will live with the consequences. And this is why many scientists and ethicists argue that genetic engineering should be restricted to therapeutic uses, preventing or curing serious diseases rather than enhancing traits like intelligence, physical strength, or appearance. But the line between therapy and enhancement is not always clear because what qualifies as a disease and where does treatment end and optimization begin? And as genetic technologies become more accessible, these questions will only become harder to ignore, especially in a world already divided by wealth, access to healthcare, and opportunity. Because if genetic engineering remains expensive and limited, it could deepen inequality, creating a future where the wealthy are not only richer but biologically healthier, while others continue to suffer from preventable genetic diseases, turning biology itself into a marker of privilege. And history shows that powerful technologies are rarely distributed fairly without deliberate effort, regulation, and global cooperation. Yet refusing to develop or use genetic engineering also carries moral weight, because every year millions of people suffer and die from diseases that could potentially be prevented or cured at the genetic level. And families watch children endure pain, knowing that the cause lies in a small error in DNA, and choosing inaction when solutions exist becomes its own ethical failure, placing humanity in a position where it must balance caution with compassion, risk with responsibility, and progress with humility. And beyond human medicine, genetic engineering also plays a critical role in controlling disease at a population level. Through genetically modified crops that resist pests, viruses, and environmental stress, reducing hunger and malnutrition that weaken immune systems and increase vulnerability to illness, and through efforts to genetically alter disease-carrying insects like mosquitoes to reduce the spread of malaria, dengue, and Zika, demonstrating that genetic engineering is not only about individual bodies, but about ecosystems, public health, and global survival. And yet every application reinforces the same lesson, that biology is interconnected, and altering one part of a system can ripple outward in ways that are difficult to predict. Making transparency, long-term research, and ethical oversight essential, because genetic engineering is not just a scientific tool, it is a decision-making framework that reflects how humanity chooses to relate to life itself. And as the technology continues to evolve, it forces society to confront deeper questions about identity, normalcy, and what it means to be healthy, because disease has shaped human history, culture, and empathy, and removing certain diseases could change how societies understand disability and difference. And while reducing suffering is widely seen as a moral good, there is also concern that genetic engineering could be used to erase traits that are part of human diversity rather than genuine sources of harm, making it crucial to distinguish between alleviating pain and enforcing conformity. And all of this means that genetic engineering and disease are not just a medical topic, but a mirror held up to humanity, reflecting our values, fears, ambitions, and capacity for responsibility. And as research accelerates, one thing becomes increasingly clear, that genetic engineering will not eliminate disease overnight. It will not make humans immortal, and it will not replace the need for social care, healthy environments, and ethical leadership. But it does represent a turning point, a shift from accepting disease as destiny to understanding it as information, something that can be read, interpreted, and sometimes corrected. And this shift changes not only how medicine operates, but how humans see themselves, not as passive victims of biology, but as participants in shaping their own biological future. At the same time, genetic engineering forces humanity to confront uncertainty in a way few technologies ever have. Because unlike machines or software, Genes interact with environments, lifestyles, emotions, and chance, meaning no genetic change exists in isolation. And this uncertainty becomes especially important when considering long-term effects, because some genetic edits may appear safe for years before unexpected consequences emerge, which is why modern genetic medicine increasingly focuses on reversible, targeted, and tightly controlled interventions rather than permanent changes wherever possible. And yet, even with caution, progress continues rapidly as researchers refine delivery systems that can target specific tissues without affecting the rest of the body, improve accuracy to reduce off-target mutations, and combine genetic engineering with AI-driven analysis to predict outcomes before a single edit is made, making medicine not only more precise but more anticipatory. And this convergence of genetics, computation, and medicine is reshaping how diseases are understood, transforming them from isolated events into patterns that can be predicted, intercepted, and sometimes prevented entirely, which may ultimately redefine healthcare itself, from a system built around hospitals and emergency treatment to one centered on early detection, lifelong monitoring, and genetic resilience. But with this transformation comes a responsibility to ensure that genetic knowledge is not misused, misunderstood, or weaponized, because history has shown how easily biological narratives can be twisted into tools of discrimination, fear, or control. And this makes education as important as innovation, ensuring that society understands what genetic engineering can and cannot do, separating realistic hope from exaggerated fear, and grounding public discussion in evidence rather than speculation. Because genetic engineering does not determine destiny, it shifts probabilities, reduces risk, and opens doors, but it does not remove the complexity of human life, nor does it eliminate the role of environment, choice, or chance. And as this technology matures, its greatest success may not be in eliminating every disease, but in changing how humanity responds to illness, replacing helplessness with understanding, panic with preparation, and resignation with agency. And future generations may look back at this era as the moment when medicine stopped fighting biology and started collaborating with it, learning to listen to the language of genes rather than trying to overpower symptoms after the damage was done. And in that sense, genetic engineering is less about control and more about alignment, about helping biological systems function as they were meant to, without unnecessary suffering caused by random mutations or inherited errors. And yet the final outcome will depend not on the technology itself, but on the choices surrounding it, on how governments regulate it, how scientists communicate it, how doctors apply it, and how societies decide who benefits from it. Because genetic engineering can either become a tool for collective healing or a source of deeper division. And that choice is unfolding right now in laboratories, clinics, policy rooms, and public conversations across the world. And as you reach the end of this video, it's worth pausing to realize how extraordinary this moment in history truly is. Because for the first time, disease is no longer only something that happens to humans. It is something humans can understand at its origin, anticipate before it appears, and in some cases prevent entirely. And that realization changes not just medicine, but how we see the human body, vulnerability, and responsibility. So if this made you think differently about health, science, or the future, take a moment to reflect on what kind of future you believe genetic engineering should create. One driven by profit or compassion, by enhancement or healing, by exclusivity or shared progress. Because the future of disease is no longer written only by nature, it is written by human decisions, and those decisions will shape not just how long we live, but how wisely we live. And if you found this valuable, thoughtful, or eye-opening, consider liking the video, subscribing for more deep explorations into science and the future, and sharing your perspective in the comments. Because the conversation about genetic engineering is not just for scientists or doctors, it's for everyone who cares about where humanity is headed. And I'll see you in the next one.